Alright, hi everybody. I'm Erin Udaley here at Roper Mountain Science Center, and today we are going to talk all about cells. And we're going to talk about how to classify cells. So we're going to basically learn how to look at a cell and decide what kind of cell it is. And the four categories we're going to work on are going to be plant cell, animal cell, protist, or bacteria. So let's talk a little bit about cells. Cells are the smallest unit of life, and all living things are made up of cells. So what that means is I'm made up of cells, you're made up of cells. If you have a pet, your pet is also made up of cells. The plants outside, made up of cells because they're alive. So everything that's alive has cells. And also, if you're thinking about the smallest thing that could be alive, that's also a cell, right? So the smallest unit of life is a cell. Okay, and speaking of small, cells are super small. They are microscopic. What that means is we cannot just look really hard and see our cells, right? We have to use a special tool called a microscope to do that. And so what we're going to do today is we're going to look at some cells through a microscope, this microscope. It is a compound microscope. There's a lens in the eyepiece up here. We have a camera mounted to our microscope so we can kind of use that to display to you guys. And down here we have additional lenses, and we can basically change and control how much we magnify by turning this little objective. And we can take a closer look. Good. I'm going to zoom us out just a bit here, and then we're going to autofocus. Okay, so this is a closer look at our microscope, and we have these three objective lenses. And whenever you're using a microscope, you want to start with the smallest one, the lowest power. The reason you want to do that is because that allows you to have the greatest field of view. And what that means is you can see the most stuff, right? And then you can decide what you want to look closely at, and then you can magnify it more by rotating to the higher magnification. Now, this low power was showing us things that were 40 times greater than we can normally see with our eyes. The medium is 100 times, and the high power is 400 times greater. So that's what we're going to be using today to look at our cells. One last thing I actually want to show you guys, though, is how to prepare a slide. Ours are prepared already, but if you guys ever get an opportunity to use a microscope and prepare a slide, it's sort of fun. Our samples we're looking today, a lot of them come from water. So the first thing you're going to look at came from this little cup. And the way we would prepare the slide is use a pipette, get a little bit of water in here, put 2 drops on our slide, and then we would take a cover slip and put it on top of the water gently, and then that would be all ready to go under the microscope. But we've got one already ready to go, so we're going to start off with that one. All right, so this first cell that I'm going to show you, we haven't talked yet about how to classify cells really, so I'm going to want you to just think in your head, best guess, what do you think you're looking at? Do you think you're looking at a plant cell, an animal cell, a protist, or a bacteria? Okay, so we're going to go ahead and take a look over here, see what we can see. Okay, so now as you're looking here, kind of try to make some observations. You can probably see movement. And we're going to move to a higher magnification in just a second. You'll see the screen. There you go. So we're going to switch into a higher power. And then I'm going to try to get us a little bit focused. Look very closely inside those little cells. See if you can see any kind of structures in there. That might be a clue. Think about their shape. That might also be a clue. Think about whether these look like these are single-celled or if they are multicellular, because that's also a big clue. We're going to the highest magnification now, and look closely. It's sort of hard on this highest power to keep them in frame. They're not super cooperative, right? So they're moving. You've got to kind of catch them with your eyes when they're going past. So kind of think in your head. The question was, what kind of cell is it? Plant, animal, protist or bacteria. Okay, great. So we're gonna leave our mystery cell for a little bit and come back to it at the end. And what we're gonna do now is we are going to talk about how to make these decisions. And we have a big handy chart over here to help us out with these decisions. So let's say you're looking at a cell and you're wanting to put it into a category. The first question you want to ask yourself is this: do you see membrane-bound organelles in that cell? And so what that means is, do you see shapes in the cell? Specifically a nucleus or mitochondria or chloroplasts or other organelles that are surrounded by a membrane. Let's say that you do not. Let's say you're looking at the cell, you don't see stuff inside of it. It seems super simple. It's also maybe super small. That's a prokaryotic cell. And prokaryotic cells, they don't have a true nucleus. They are super simple. So let's look at our options here. Which of these do you think is the smallest and the simplest? Think a guess in your head. If you're thinking bacteria, you are correct. Bacteria are prokaryotic cells, super small, super simple, no nucleus. That's them. Now, let's say we were looking at our cell and the answer was yes, we saw some structures in there. That tells us it was a eukaryotic cell. All of these cells are eukaryotic cells. That means they have a true nucleus. Okay, next question. Is it unicellular? Is it just one cell? Like a unicycle has just one wheel? Is this thing just one cell? If the answer is yes, what do you think? Which one of these do you think is one cell? Hopefully you're thinking protist. So protists are single-celled eukaryotic organisms. They do have those organelles in them, those membrane-bound organelles, but they're just made of one cell. All right, let's say you were like, no, it's not unicellular. I see a bunch of cells, or I know it came from something with a bunch of cells. The next question you ask yourself, can you see cell walls? Can you see chloroplasts? Chloroplasts are, that is where photosynthesis takes place. There is chlorophyll in there. This is how a plant gets energy, right, from the sun. And cell walls are kind of like, kind of give a set structure, right? So a lot of times these kinds of cells look very regular in shape. I bet you know we're talking about a plant cell, right? Because I said photosynthesis. I think I also said plants. All right, so plant cells. Let's say the answer was no cell wall, no chloroplast. It looks kind of blobby. Looks like it just has a cell membrane around it. That's us. That's an animal cell. So that's how you're going to make these choices as we go through. All right. Awesome. Okay. So now what we're going to do is we're going to take a look at our first cell type. And we've got everything already prepared from here on out. So we're going to kind of take a look. And this is under low power. So let's go ahead and start trying to make some observations. And we're going to, I'm going to zoom and focus and get this to where hopefully you can see really well inside. Remember your questions. As we zoom in here, think about, can you see structures inside of the cell? If you can, that's telling you something important. It's telling you it's eukaryotic. And then the next question you're supposed to be asking yourself is it single-celled, or does it seem like it is a multicellular thing? There are many cells together. And then the next question is, do you see chloroplasts, which are going to appear to be green? And do you see cell walls, which will be kind of give like a geometric shape almost, like a sort of a nice predictable sort of shape that seems pretty consistent. And while we've got this slide, if you look closely, you might even see a little bit of movement. I can see a little bit of movement in the cytoplasm. Cytoplasm is that sort of gel-like stuff that is inside of the cell. So it's kind of interesting that you can see it moving because it is actually alive. Okay, so think in your head, what was that I was looking at? Hopefully you were thinking plant cell, because it was a plant cell. And there were many clues there. The main one was you saw cell walls. You saw that nice, like, it was like rectangles. You saw chloroplasts, which is another really good signal because you saw that bright green color. And you also, when you saw those chloroplasts, those are organelles. Those are membrane-bound organelles. That was a signal that you were looking at a eukaryotic cell. And you saw many cells stacked right next to each other. So you could probably tell you were looking at something that was multicellular. All right, let's take a look at what you were looking at actually. Hold on one second, let me adjust this guy. Okay, good. You were looking at this plant. We were looking at one leaf. This is called Elodea. It is a plant that lives in the water, and it's sitting here in a little bit of water here. So that's what you were looking at. It's got super thin leaves, so you can see the cells really nicely, even like while they're still alive. All right, good job. Okay, we're actually gonna look, so this one I'm not gonna ask you to think about what it is. I'm gonna tell you it's another plant cell. I want to look at it because it's a little bit different from the Elodea. On this one, this is an onion. These are onion cells. They are stained. We purchased them, and they're stained so that you can see that nucleus. If you notice that round structure inside, that's the nucleus. We weren't able to see those on our Elodea cells because of all those chloroplasts, and that one wasn't stained. So just so you're aware, those plant cells do have a nucleus as well. And since this is an onion cell, think about onions, they're underground, so not having to do a lot of photosynthesis, that part of the plant. All right, good job. Okay, time for the next one. So this is going to be a new cell type, and you guys are going to try to decide in your head what you think you're looking at, and I'm going to give you some hints while we look. Okay, so this is a prepared slide for us. So we purchased this slide, which is why it's so colorful, because it is also stained. You've got that dark purple color. Here's a hint. The things that are dark purple, each of those is a nucleus. And the pink that you can see, let's get in focus here, that pink color is showing you the cytoplasm. Take a look at the edges of the cell and try to decide if you think it looks super regular or if it seems like it's a more flexible layer, less rigid, because that's also an important signal. And think about whether you think this is a single-celled or a multicellular thing that you're looking at. Okay, now hopefully, remember your choices: plant, animal, protist, bacteria. Hopefully you were thinking that that was an animal cell, because it was. That was actually, those were actually liver cells from an amphiuma, which is that creature. It's a salamander that lives in the water. Again, we did not prepare that, so we didn't hurt any amphiumas. It did have membrane-bound organelles. There were those nuclei you were able to see very bright. So it was eukaryotic. You could see a bunch of cells together, so we knew it was multicellular. There were no cell walls. It was more blobby, and you didn't see any chloroplasts either. Good job. All right. Now we're going to look at another animal cell here. This is actually going to be a cheek cell from a human. So we did not prepare this one. We purchased this one. But this is a cell that was scraped off with like a toothpick of somebody's cheek. So it looks like it's single-celled, but it came from a human. That darker circle inside, that's the nucleus in there. Good. So that would be what a cell from your cheek would look like if you were to stain it. All right, doing awesome. Okay, next one. And this one is a sample. We also purchased this one, and it's also been stained, so it's that bright pink color. We are already at our highest power here, so this is about as magnified as we can get. We can zoom in a little bit with the phone. So those little pink shapes, this is a sample from stagnant water. So try to think in your head, what do you think you're looking at? Plant, animal, protist, or bacteria. And this is about as magnified as we can get this. So think about if we can see structures in those cells or not. Okay, so think in your head, what do you think you're looking at? Super duper small, super duper simple. Hopefully you were thinking bacteria. That's what that was. That was bacteria that was stained. If it wasn't stained, we probably couldn't even see it. And we couldn't see it very well, even with our microscope. So, right. So we would need even more powerful microscope to really see the structures inside of that bacteria. Okay, let's look at our last one. Okay, good. Take a look here. Ask yourself, what do you see? Does it seem like it has membrane-bound organelles? Look super closely inside of it. Try to, do you think it's multicellular or single-celled? And try to make some observations about its movement. And Remember your choices: plant, animal, protist, or bacteria. And this guy's moving around, so it's a little bit hard to keep him on the screen. Okay, hopefully you guys were thinking protist, because that was a protist. It does have membrane-bound organelles. It's single-celled. That was a paramecium you were just looking at. And they have little cilia around them to help them move. We really weren't able to see those with our microscope, but eukaryotic, single-celled. This protist group is very diverse. It includes amoebas and also euglenas. And okay, that being said, let's go back to our very, very first cell that we looked at here. Okay, so remember you were supposed to try to decide what you thought these were. Now, we're going to make some observations really quick. We can see movement. They appear to be single-celled. We're going to zoom in a little bit closer in just a second here. And when we do that, hopefully you'll be able to see some structures inside. So a lot of those are giving you kind of some hints here. Okay, guys, so you took another look at our mystery cell there, and hopefully you thought maybe it was a protist, because that is what it was. It was a very confusing protist, though. It was actually one of these euglena. They are tricky because they have chloroplasts. You may have noticed that it was green, and you were able to see those little organelles in the little cell body there, but it actually was a protist. So if you thought that, awesome job. Now I want to just take a second and show you guys where our euglena came from. Oh, if we could switch to the Elmo camera. I think it's number 3. There you go. Good, thank you. Okay, so let's look at this guy. Actually, this is just water, right? But it looks green, and it looks green because it is full of euglena. Good. And actually, we can just stay on this view because I also want to show them where our paramecium came from. It came from this container. So this is where we got them from. They're in there. There's food for bacteria, which is what our paramecium is eating. Okay, great. We can go back to the main one. Awesome, thank you. Okay, guys, so great job classifying cells today. Hopefully you feel a little bit more confident in deciding if a cell is a plant cell, an animal cell, a protist, or a bacteria based on their characteristics. All right, thank you so much. We can't wait to see you here at Roper Mountain again soon. Have a great day. Bye.